We are unable to physically enter the brain, as the blood vessels are heavily secured and guarded by a layer of epithelial cells called the blood-brain barrier. We can, however, create a virtual model and simulation to display the myriad of cells in the nervous system. Nerve cells are roughly grouped into two types, neurons and glia. The main difference is that neurons control the body functions and are responsible for every thought, memory, and feeling we get. Glia are cells that support, care for, and protect neurons. Astrocytes are a type of glial cell and look something like this, with many branch-like appendages called processes. When most people think of the nervous system, they think of neurons. However, astrocytes are about the most numerous type of nerve cell in the human brain. They have many functions. These include providing most of the structure in the nervous system, creating glial scar tissue to block off damaged tissue, and maintaining a balance of charge in the interstitial fluid that surrounds nerve cells. The importance of this last one will be explained later. They also care for neurons, giving them lactate and clearing out neurotransmitters from their synapses. The last major role they play is controlling the diffusion of molecules into the brain from the blood vessels. A second type of glial cell, microglia, actually come from monocytes. The main purpose of microglia is to destroy damaged cells and invading pathogens. They have long processes like astrocytes, which they use to monitor the interstitial fluid. They are extremely sensitive, and when they detect an abnormality, they retract their processes and become activated microglia. In this state, they seek out the abnormality and remove it as best they can through phagocytosis, the process of engulfing something and digesting it. They cannot continue this forever, and they eventually degrade into a non-reactive cellular mass. The third type of glial cell is the ependymal cell. This cell lines sinuses and provides a barrier between the interstitial fluid and the cerebrospinal fluid, the liquid that absorbs shock to protect the sensitive neurons. What we see here is a typical neuron. It has a soma, or the main body that contains all the organelles, dendrites, which are the branch-like projections at the top of the neuron, and the axon, the long extension that exits the soma. The dendrites of the neuron are for receiving messages from other neurons or other stimuli. They receive a signal called an action potential, which will be explained here. An action potential is basically an unbalance in electrical charge that travels through a neuron. It is the way neurons communicate with each other. There is normally a more negative charge on the inside of the neuron compared to the outside. Two types of graded potentials, or charges, come down the dendrites, inhibitory and excitatory. Inhibitory are negative charges that make the threshold potential, the point where a signal is sent down the axon, less likely to be reached. Excitatory charges are positive. They bring the total voltage closer to the threshold potential. The threshold potential is the point where the difference in electric charge from the inside and the outside of the neuron is about negative 50 millivolts. The resting potential, or the normal charge, is about negative 70 millivolts, which means the inside of the neuron has 70 millivolts less of electric charge than the outside of the neuron. This means the excitatory and inhibitory graded potentials must add up to raise the charge inside of the neuron by about 20 millivolts for the threshold potential of negative 50 millivolts to be reached. The graded potentials are added up at the point where the soma meets the axon, called the axon hillock. If they add up to the threshold potential, proteins in the cell membrane flip open, allowing positive ions in. They close after the positive charge gets to a certain point, and the positive charge dissipates down the axon. The coverings on the axon are myelin sheaths, made of cells called oligodendrocytes and Schwann cells. Oligodendrocytes have long processes that end in myelin sheaths, and they can provide more than one section of it. They produce the myelin sheath for neurons in the central nervous system, or the brain and spinal cord. Schwann cells create the myelin sheath by wrapping their entire body around the axon. They only provide one section of myelin, and they produce it for the peripheral nervous system, or the neurons outside of the brain and spinal cord. The myelin sheath is an insulative covering that prevents the electrical charge from escaping from the axon wire. The gaps in the myelin sheath are points where the electrical signal is boosted. Just like at the axon hillock, when the positive charge hits certain proteins, they let in positive ions to boost the signal to a certain voltage. After a positive charge passes, other proteins restore the amount of negative charge inside and positive charge outside the axon. This is how neurons send signals to each other. The axon ends in a synapse, a gap large enough to stop the electrical signal from traveling further. The electrical signal does, however, stimulate packages of chemicals called neurotransmitters, symbolized by the particles, to be released into the synapse. They bind to receptors on, for example, a dendrite on the neighboring neuron. This causes a graded potential to be sent down the dendrite, and the whole process is repeated again. Any neurotransmitters that get lost are soon cleaned up by astrocytes.